Okay, so I would like to introduce the first speaker for the skin and musculoskeletal tissue session, Dr. Anthony Oro. He is a professor of dermatology from Stanford University. Is Dr. Oro here? Yes.
Okay.
Can you, please.
Thank you very much. Thanks for great, super, and slides. And perfect. Thanks. So, thanks. Lawrence gave an amazing introduction, and as far as the skin viewpoint as a physician and dermatologist, I really wanted to highlight and emphasize some particular points in terms of developing skin therapy. This is an example of the skin. And the skin, as you know, is an ecology of cells, which include the keratinocytes up here in the bat, which are the star players to this morning, which provide a barrier to the outside world. But it's an ecology of cells with pigment-forming cells, hairs, melanocytes, fat, and so it's this ecology that provides together. And so when you think about the tissue, you have to think about it all together. And with cell therapy, we often think about it as individual cells. And so that is something that we have to think about in terms of overall organ function versus just cell therapy together. That's one point I wanted to make this morning. Now, skin is accessible, and so a long history of autologous and allogeneic cell therapies in skin, dating back from skin grafts from people that cut off their nose and they wanted to graft over 4,000 years ago. Many of also autologous follicular units, hair transplants, which are just really redistribution of autologous hair units. And then even melanocyte transfer for vitiligo, especially in patients who have this, are also being done. Now, the problem here is that these are all, as Lawrence, and an issue we're going to have to address today is scaling autologous tissues. Because if a hair transplant works, but if it's too far gone, you don't have autologous units to graft, you're out of luck. And so the real question is, or if you have a tissue or a disease such as epidermolysis bullosa, shown here, where you have a defect in one of the 18 proteins that keeps your epidermis onto your skin, or you're a combat veteran that has extensive burns, you're out of luck because you can't surgically transplant it, you can't give them a drug, and so you need to regenerate the tissue in some way. And that's the issue we're trying to address with regenerative medicine in terms of scaling autologous tissues. And so the rest of the time I'm going to talk about today is about our work with recessive dystrophic epidermolysis bullosa, which is a collagen 7 deficiency, one of the 18 proteins, and our efforts to generate autologous skin grafts for these patients. So because it's a horrible disease, where these patients will basically often die at birth, or if we actually manage to keep them alive, we wrap them like mummies for the rest of their life. So palliative therapy is the only thing you can do. And then if they actually live long enough, what happens is that they die of invasive squamous cell carcinoma because of the chronic wounding and mutations that they get. So because of the horrible nature of this disease, the entire field, and anybody who is trying to develop therapies for this in terms of massive production of protein and then using bone marrow transplants or allogeneic mesenchymal stem cells, what I want to talk about today is the work that we've done in keratinocytes, because it sort of illustrates the issues which we're trying to address in the symposium today. And that is the development of an autologous retroviral-corrected sheet product we call LEES, which is really taking an autologous keratinocyte, which, as Lawrence talked about, I'll show you the data, is the target cell that's transplantable. And so that's from, again, 4,000 years of skin transplantation. We know that the keratinocyte is this, and that's actually, we're lucky that way. But that's, even though we had 4,000 years of research, we still took a long time to develop the product. And so this is the proof of concept trial, where we take a skin biopsy of the patient. We then, for the affected keratinocytes, we can culture them because of 40 or 50 years of burn literature, burn research. We use an old-fashioned Maloney retrovirus that carries the gene and inserts it into those keratinocytes, make 6 skin grafts, and then graft them on the patient. We're not trying to cure the whole disease. We're just trying to do proof of concept on these patients in a phase 1 clinical trial. Now, that sounds really easy. However, the initial, as again, another theme here is patience, because the initial preclinical data showing that this worked was done by Paul Cavari in 1996 in a Nature Medicine article. However, the phase 1 clinical trial, which our group had just done, started in 2014. That's 18 years. So what happened in that 18 years? And it was all academic development, because at the time no one really had the technology or the availability or the know-how to actually do this process development. Okay, so that 18 years, and we can talk about the various issues in terms of funding, trying promotion, academics, and that's the issues that Lawrence talked about. But the phase 1 clinical trial was done, and fortunately, it actually was very successful. It was just recently completed, a 2-year phase 1 clinical trial, coming out next month. It's in press, but really taking 4 patients, 6 grafts each, grafting on autologous corrected facilities in GMP-like conditions. And you can see both safety and efficacy was met, where these 24 grafts, now actually out 2 years, were very effective. So this really shows that the keratinocyte stem cell, when corrected and produces enough collagen 7, can actually have very impressive and remarkable, actually, disease-modifying activity in these patients. So that's really exciting. But the issues then going forward are, again, going back to this issue of autologous scalability. These patients have low keratinocyte stem cell numbers because of chronic wounding, and so trying to graft the whole body or to take to turn them into GMP-grade facility products are challenging. Gene transfer using the jalopy Maloney retrovirus doesn't work for dominant negative diseases, and safety concerns of permanently integrated retroviruses. And also these patients, because of their chronic wounding, have preclinical, pre-malignant lesions in their stem cells that we're grafting. And so if you graft them and they're going to get squamous cell carcinoma in a few years, we're giving them a squamous cell carcinoma, predisposed keratinocytes. So these are all issues which we're trying to deal with going forward in terms of the scale of the target product profile you want to give clinically to the patient. And so in collaboration with Marius Werning, my longtime collaborator at Stanford, and in this crowd, this is not unusual, we used to use IPS cells, these induced pluripotent cells, which we can use in terms of a process called therapeutic reprogramming, where you can take the skin biopsy, use the Yamanaka reprogramming factors to make induced pluripotent cells for these patients, and then use genome editing techniques to correct the particular disease. And now you have a iPS cell for the patient, which is corrected, and then you can then use downstream differentiation techniques, which Lawrence talked about, to make any tissue in the body that's defective, in this case, human skin. We actually then use the sequencing technique to actually clone out some of the bad, the cells that have bad mutations, which I'll talk about in a second. But this therapeutic reprogramming technique, because you can scale the iPS cells, is actually a very nice way to make a cell bank for the downstream production. So in this case, we use iPS cells. Lawrence talked about direct reprogramming, but in any case, when you think about the cell, you want to have a defined cell that you can scale, that you know is safe and efficacious. And there's direct reprogramming techniques, but really the iPS cell is one of the ones that actually you can use because you can grow them and scale them. So what are some of the issues to talk about that we've learned from using therapeutic reprogramming for scaling? The first one is that iPS cells are very nice for a cell bank for scalability and for safety and efficacy, because you can actually take it and define it from a single cell. And so with our EB patients, about half of them will die from overwhelming squamous cell carcinoma. Because of a chronic wounding, they get squamous cell carcinoma mutations. So what can you do about this? Well, we know that each iPS clone that we make comes from a single cell. So if you take a bunch of clones and then sequence them for squamous cell carcinoma predisposing mutations, you can then pick out the good ones and leave the bad ones in the dish. And that's exactly what we did. The other part of my lab works on skin cancer. And so we've done the entire mutational analysis, along with Paul Kovari, for all of the keratinocyte-derived diseases, skin cancers in humans. I work on basal cell carcinoma, but for both basal cell and squamous cell carcinoma, we know the genes that predispose to mutation, to cancer. And so you can develop very nice beads that can actually sequence the individual clones. Here's 3 examples of those clones, and you can see that these 2 actually have concerning mutations in them that would predispose, notch mutation that would predispose to squamous cell carcinoma. This particular one actually is very nice, and this is the one you choose for downstream therapeutic applications. Issue 2, which we actually is we actually can use genome editing techniques. At the time we started this, we used recombinogenic AAV. You can use CRISPR-Cas9 now, and the genes in the genome should we be editing for using these for therapeutic applications. Our patients have a mutation in collagen 7, so it seems it's an easy ethical issue to actually then just correct that and then go on in life. But what about other things, correcting other issues? Actually, National Academy last year had a whole summit on human genome editing, and this is something we need to address in terms of for therapy in the future, for cell therapy. What should we be editing besides the actual disease mutation that these cells have? And then in terms of taking these cells that are iPS cells and then trying to make graftable products, for example, for autologous skin grafts, we developed a very nice skin tissue bioengineering protocol where we can take patients' ES and iPS cells and make keratinocytes that then, when you graft them onto an animal model, you then make a very nice human skin that stratifies and has, protects. But the issue is safety and efficacy. Here you can see that if you take individual iPS cell lines from the same patient, doing multiple different runs, you see a wide variety of the ability to make in the same assay the same keratinocytes. Each individual iPS clone actually has, because of epigenetic reprogramming, has different abilities to make, and so we have to understand how to do this, how to make the manufacture a defined product from different clones. And so the problem is, if you want a self-driving car, like the Google self-driving car, what you would need to have is first is the map, is a Google map. And so what we need is a map of differentiation. We need to know how the chromatin folds, how all the epigenetic marks work from the start to the finish. And so what we need to do, and what we're doing now, is basically using multigenomic genetics analysis, RNA-seq, ATAC-seq, ChIP-seq, Capture-seq, to understand how every single base pair is folding in these cells from start to finish, to then to have in-process biomarkers that are relevant to the differentiation product to manufacture them. This is so that we know, if you're like making where you are in terms of the Google map, you know where you are in the world. Okay, and lastly, I just want to point out that consortia, as Lawrence talked about, are critical. We now formed an EB consortium with different groups. Each of us have made these iPS-derived keratinocytes a different way, and we're getting together to form a coordinated process. I think going back to why we took 18 years in the beginning, one of the reasons is because we did it by ourselves. Friends help you if you can work together. And then at Stanford, we've developed a Center for Definitive Medicine, where we actually can take multiple different programs of cell therapy and our new GMP facility at Stanford to try to then use best practices within each different organization and therapy to then try to accelerate it. We want to avoid taking 18 years again to develop the next product. And with that, just to summarize, skin is a rich history of cell therapy for autologous diseases. For genetic diseases like epidermolysis bullosa, autologous corrected keratinate sheets have powerful, long-lasting disease-modifying activity. iPS cells and genome editing can have a promise, and consortia and working with your friends and best practices, I think, will go down from 18 years, hopefully, to a much shorter time. So thank you.